Good morning, everyone, and welcome to the dkNET 3.0 Introductory Webinar. This webinar is being recorded, and we will be placing this webinar along with our others that are already posted on our YouTube channel. So in case you've missed one in the past, you can check out the YouTube channel and catch up on some of the other webinars. And today, what we will be covering is the recent release of dkNET. And let me see, I need to get my slides moving here, which is, oops, let's see. Okay, there we go. Somehow my keyboard isn't working. So the outline for today's talk is we're going to have a brief introduction of dkNET, then get into the four main areas of the new dkNET 3 release. The first is the Resource and Information Network, information about research resources that will assist you in making informed decisions about various research resources such as model organisms, antibodies, software tools, and the like. Then we'll move on to a section where we'll cover our authentication reports, and this is to help researchers comply with some of the NIH mandates in terms of submitting proposals and also submitting progress reports in terms of information about authentication of key biological resources. Then touch on briefly on fair data services and the Hypothesis Center. So dkNET was launched to serve needs of basic and clinical investigators in the areas supported by NIDDK, so diabetes, digestive, endocrine, metabolic, kidney, and urologic diseases. dkNET started by providing seamless access to a collection of diverse research resources. So dkNET sits on top of an aggregation of data, information, materials, information on funding opportunities, literature that advance the mission of NIDDK. And what we have been doing in this latest round is sort of been expanding those resources to provide tools and services in support of increased rigor and reproducibility. And really trying to assist researchers to improve science and the reproducibility of science. So dkNET 3.0 ties together a number of aspects related to improving dkNET in terms of providing information really targeted towards this rigor and reproducibility. And so those four areas, again, are the resource reports, the authentication reports, the fair data services, and the Hypothesis Center. And one note is we've just released 3.0 as a production release, but we still will be making frequent updates and improvements. So stay tuned to the newsletter, where we'll announce sort of major updates to the system as we move forward, because this is continuously under development. And please give us feedback if there's anything you'd like to see, or if you have any comments in terms of how we could provide this information in a more useful fashion. So the discovery portal is the base of dkNET in the sense of this is what brings together a lot of the information that we build our other tools and features on top of. And so the discovery portal connects researchers to more than 300 biomedical databases in terms of aggregating that information and making that searchable from a single user interface. And when you search, as you can see on the left, you will be given an information as to what types of data are being returned. You can then sub-select various areas. You'll get a listing of the databases that have that material. And then you can actually go into a single resource, such as the NIDDK Central Repository shown on the right, that provides information on information in that repository. And then just like Google, we link you out directly to that repository where you can actually gain or grab that information and delve a little deeper. And we provide services on top of this as well. So in addition to doing this, as a user, you can also do this programmatically, incorporate, those results in tools that you're developing. So built on top of this ability to aggregate information, we've been aggregating information around research resources, and a key issue has been that key biological resources are sometimes very difficult to find in the literature. And so, this is an example where we have a model organism from Jackson Laboratories. When you go in and you actually, try to search, within, Jackson Labs itself, the text used to describe, the model organism does not yield a very good search result. And this is, in terms of, how we actually describe things in the literature versus the way things are described in the databases. And so one of the things that we looked at a number of years ago is how could we make this easier for researchers, to basically document what type of resource they're using. And this is where the RRIDs, the Research Resource Identifiers, and the pilot project for this were born, in a sense that, we can actually provide, simple identifiers for research resources in the literature, given that a lot of these are provided and maintained in terms of their catalog lists by databases. So the model organisms are maintained by a number of model organism databases where they have proper identifiers. For the antibodies, we have the antibody registry. For cell lines, there's Cellosaurus. So there are a number of these community sites that manage the identification of various resources. And so by pulling that information together, we could easily provide the ability to identify resources in the literature. And this is now being supported by many major publishers, including Nature, the Cell journals, Endocrinology, and there are now more than 700 journals with RRIDs, and many of these journals have information on RRIDs in the author guidelines. Some of the journals actually require this information to be provided, and so there's now a growing number of these well-identified resources in the literature available to researchers. And there are two primary reasons, why RRIDs were created. And one of the first was the issue of tracking, right? So who is using various model organisms, who's using various antibodies, and if you're actually tracking these in the literature, you can actually then see, their uses, how is the use in a certain experimental paradigm, and also be able to tie into potentially who else is using these resources in case, one has questions around that. The second reason relates to rigor and transparency. So being able to provide information on these resources, what their application has been, and to basically create an environment where we have information on cell lines, for example, where you have curation being done via Cellosaurus and the Cell Line Authentication Committee, where people are looking at the literature and then annotating information in this community authority around cell lines, providing documentation, if a cell line's been discontinued, if a cell line's been contaminated. So by having these IDs that tie this information together, it makes it very easy for someone who's looking at potentially using a cell line then to tie into information, about its applicability and its current status. And so with the advent of the RRIDs, we've gone back and we've looked. So there's a paper from 2015, where the identifiability of these resources has improved dramatically. And that also lowers the cost of curation in terms of discovering where these resources are being used. And so our resource reports build on these RIDs and provide information to researchers, basic information about an antibody, for example, in this case, but then also usage and citation metrics, and also information about a potential collaborator network. So who is using them? Does an antibody have any issues? In the future, one of the features we'll be adding is how do they compare, right? So in terms of information we've gleaned from the literature, providing comparisons between a number of antibodies. And then also, is anyone using them near me? So is there anyone near me who has any experience with this resource? And so when one comes to the dkNET site, if one clicks on the resource reports, in this case, clicking on the antibodies, one is taken to a search page where one can type in keywords that help you narrow it down, but we also provide resource-specific fields, such as a vendor and catalog number, to really help you easily find, antibodies that you may be using or may be thinking about using. Once you actually find an antibody and you select that antibody, you'll be taken to a specific page with that antibody. In this case, what we're showing is a table view. So if you switch between the antibody and the snippet view shown on top here and the table view, there are two different ways in terms of how we display this information. A table view is provided to make it easier for people to sort and filter among a number of different records if more than a few records are being returned, which sometimes can be the case. And then once one gets to the actual resource summary report, again, we have the basic information. This resource information, as I said, is updated from these community resource registries. So currently we have 4, which is antibodies via the antibody registry, cell lines to Cellosaurus, research resources such as software, databases, through a tool registry, the SciCrunch registry, and then the organisms, the model organisms, come from a whole host of organism repositories. And when one looks at this report, in this case for this antibody, we can see that it's been cited in 204 papers. We give some of the latest papers that we have found with this. And so one thing, in terms of how we actually pull this information is that we actually mine the literature. So we mine PubMed abstracts, but we also mine various parts of the open access literature that are available to being crawled. And so that includes PubMed Central, their open access subset, and then some journals that we have agreements with to do this text mining. So we pull those RIDs out, and we monthly make those available. So we have monthly updates on these citation metrics. And when one actually looks at those citation metrics, one has a number of different things that one can see. So if you look on the right, you'll notice that we have a usage graph that shows how many articles per year in terms of this resource, in terms of it being used in the literature. We have other information talking about where we're getting some of this information in terms of the text mining. We also list the top 5, up to the top 5 other research resources that are frequently mentioned with this resource. So for example, antibodies might be other antibodies that are used together for software tools. A lot of times what we may see is that a software tool, for example, for analysis is listed along with some software tools for visualization, as well as the software, that software tool was written in, for example, MATLAB. We then provide additional report filters where one can look again for researchers that may be near you in terms of use of this resource. And so if you type in that information, what we're given with is, so for example, if we're looking for, in this case, California, we'll return information about researchers who have published using this resource and where they're located. And again, this is one of the newer areas of the portal, and so we're continually adding more location information. So this information will also be built out over the coming months as we increase the number of locations we're extracting from the literature. The last and probably most important part of the resource reports is the area that focuses on ratings and alerts. And to enable this, we're aggregating information from a number of places that provide validation information for antibodies. So for example, ENCODE, Human Protein Atlas, and other resource centers are providing validation information. So we're using the tools that we have to aggregate information, we're pulling that information together, and then trying to make it easily available via these resource reports, so that you can find that there is validation data available for a certain antibody, for example, here that's available via Atlas antibodies at the Human Protein Atlas. For the ratings and alerts above that, we show information on a problematic cell line. And so again, we're trying to make these easily visible towards the user when they come in and look at one of the reports for one of the resources that they're using or planning on using. Sometimes you may have a resource that's actually not registered somewhere yet. So for example, if it's a cell line that has been developed by your lab, one of the things that we will actually encourage you to do then is, if you're looking for resource reports or using some of the other tools, is to actually then register your cell line with Cellosource or your antibody with the antibody registry. And for many of these, registration is fairly quick. So for the tool registry, antibody registry, these are fairly easy for you to register. It could take a couple days. Cellosource also has a good turnaround time, and then the model organism databases depends on the model organism. Who you need to speak there in terms of providing that information. One of the things that we at dkNET are also working on is we're also looking at ways that we can add additional types of resources to this RID environment. And so most recently we worked with the Integrated Islet Distribution Program and the biosamples databases here at NIH, at NCBI, and also in the EU, is to develop a means to provide identifiers for these pancreatic islets used by IDP researchers, in a sense creating these RIDs for biosamples. And so we've worked out that pathway, working closely with a number of partners at the IIDP and also with the Human Islet Research Network, HERN, to develop this information system to allow this documentation of biosamples. And this is the newest, one of the newest areas that we've been working on, and this information on these biosamples will also be made available via the dkNET portal in the near future. So building on top of these resource reports, now that we have a lot of this information about these research resources, how can we make some of the requirements for researchers that NIH is mandating in terms of proposals and the like? How can we make tools to assist researchers in managing some of that information? And a lot of this has to do about authentication. So when one looks at cell lines, for example, there are a number of standards in terms of what's suggested in terms of authenticating your cell line. One of them is clearly, check if it is a known contaminated cell line. But again, researchers, may not know exactly where to go to check. You can't just go into the literature a lot of times and do a search, because there are a lot of different papers, and tracking down that information may not be that simple. And it still is an issue, right? So we do still publish papers with contaminated cell lines. So there's articles about this. And most recently, there was a paper that came out this year where we're looking at papers where an author had to use an RID or get an RID for a cell line, versus papers where we auto-detected, where we basically did some text mining to find cell lines within the papers that didn't have an RID associated with them. So comparing, the papers that had weren't required, or they didn't use RIDs at the time, and papers where authors are using RIDs, we see much lower usage of lower reported use of problematic cell lines in that biomedical literature where researchers were using RIDs. And so that's an important guide in terms of saying that RIDs do help in this. But one of the things that is most important is we don't want to do it only at the publication stage of research, right? So if you've already done all your research and then you're doing your publication, you have to find your RID, and then you end up on that resource page and find out that your resource is problematic. You've already invested a lot of time and effort in your actual research, right? So you don't want to go through the whole research process and then at the end find out, through some of these databases and information resources that have this information, that you had a problematic resource. So what we want to be able to do is enable RIDs to warn authors before publishing, right? And so again, within our system, whenever you're doing searches on resources, whenever we have information on problematic cell lines, we're showing that information to you as a researcher, as soon as you start, exploring these resources. And as part of new NIH proposal guidelines, researchers are meant to provide a plan on how they're going to authenticate their key biological and chemical resources. And so this is a separate section within a research proposal. And there are a number of sort of pillars for validation, for example, in the antibodies. So there are a number of guidelines that have been provided. And so this information is some of the information that we provide on our site, to assist researchers. We point back to these original publications, but we provide an information resource around the authentication of antibodies and cell lines. But in addition to that, we also provide the ability to create these authentication reports around the resources that a researcher is planning to use. So a researcher can come to the site, can collect the information about the resources that they're planning to use, and also get information about means of authentication for those resources, and then links to the primary publications that describe those methods. And so a custom authentication report, we have a dashboard where one can create these authentication reports. And once you create a report, you can find your resources and you can add them to your basket. So on top right here, you'll see the items in your basket. And again, here, just for illustrative purposes, we've selected a number of resources that show potential problems. And again, if you look down on the screen where we're doing the search, wherever we can, we provide warnings and feedback about resources and if they're problematic. Once you have a report, you can preview that and save that. And what you get back is a PDF document, an HTML document, which you can also snapshot to a PDF document, that basically has information on sections on the cell lines, on the antibody. So in each section, there's information on the actual resources that were included, some basic information about that resource, and then also any warning information. We also provide then information on what the community suggests in terms of authentication, and then we provide the citations for all that information. So for the resources, but also, for example, they were taken from Cellosaurus, but then also information, for example, here, the cell line checklist for manuscripts and grant applications, and then standards for cell line authentication. So we provide this information to the researchers to make it easier for them to generate their authentication plans. And for the antibody section, it's the same basic ideas that we have the information on the antibodies. We have the validation information, which here includes the number of different pillars based on how you are using that antibody. We also provide a link to information about model organisms. So for example, in the genetic pillar, looking for a specific model organism based on some gene disruption or knockdown, potentially you can find organisms through dkNET as well related to that. And so again, we're trying to make this information easily available and provide a simple way for people to also track their resources. So for example, you may be using a newly developed cell line that you could add to a resource report. Once it's saved, you'll have continued access to that, so that at some point, 6 months, a year down the road when you're preparing a publication, you can always come back and see if additional information has been found for that resource. So providing you sort of updated information about the resources you're using. And this is also one area where we'll be providing some automated notifications in the future as we further develop the portal. The last part of the authentication report is the catalog, the sort of the resource index. And this provides, a breakdown of all the information that we have, from these various community registries for these different resources. So it provides you a nice little index with the information from those databases in one report. So it aggregates all that information. So these, the resource reports and the authentication reports are really the sort of the two main products that we're making available through the portal to assist in the use of research resources by investigators. Another area that we're focusing on is FAIR data services. So FAIR, for those who may not know the acronym, is a set of high-level principles to ensure better sharing of data within the research community. So high-level principles, to make sure that data are findable, accessible, interoperable, and reusable. And so these principles were published in 2016, and this was an activity that was really brought together by the Force 11 community, the Future of Research Communications and E-Scholarship. But the main idea for the FAIR principles is really to assist in the discovery and reuse of research data by third parties. So it's been recognized by major initiatives in Europe, through Elixir and Horizons 2020, and also, in the US through the National Institutes of Health and the National Science Foundation, but really making sure that data generated, research generated via, public funds is available to the research community. And so this is a big drive, as I said, within the US and in Europe. And to assist researchers, in terms of making their data fair, we're starting to provide a number of different resources. So we have some information on best practices for managing research data. We've compiled the list of community-approved repositories, where someone coming to dkNET, can look at this list of repositories to see if there's something appropriate for their data. And we're also providing, information, one-on-one. If you do have questions about, your data or, what you might be required to do with your data, you can always get in contact with us and we'll try to help you, in terms of the various requirements that you might be mandated to adhere to. We're also developing a tool for creating a fair data plan. So the idea here is to have a more automated tool where we can ask a number of different questions, and those questions will then guide you through the process of, what you should be doing as part of data management, and then also as part of deposition of that data once the research study has been completed. So building on top of fair data, so once we have data that is accessible and also curated in an ability to be used in a uniform fashion, we also are building out what we are calling the Hypothesis Center. So this is a partnership between the dkNET team at UCSD with the Signaling Pathways project led by Neil McKenna at Baylor. And the real goal of the Hypothesis Center is to provide tools to allow researchers to easily sort of mine and explore data, for the use of hypothesis generation. So the Signaling Pathways project enables this across omics data. For example, a lot of the omics data created in Geo. However, data in Geo, if one has ever interacted with Geo, a lot of the data is not very clean, so it makes it very hard to right off the bat combine this data and explore that data together. And so what the Signaling Pathways project does is it does fair data curation on top of the data within Geo to make sure that this data can be interoperable. And once that data is interoperable, they're able to put together this knowledge base, which allows researchers to easily explore, the various role of genes and other types of omics data in terms of how they affect various signaling pathways, how they might be involved, in various types of disease. So really allowing you to explore this genomics data in an easy fashion. This is another area, this is a new area for dkNET 3, and will be continue to be developed. And so again, any feedback that you have on this section, please let us know. But those are really the 4 main areas within dkNET. We will be having a much more detailed webinar on authentication reports and rigor and reproducibility on Friday, May 24th, so in 2 weeks. So I would invite everyone to come back and check that out. And otherwise, I am done with this presentation. So if anyone has any questions, you can use the chat box, or if you have any questions, if you can, I'm not sure if you can unmute yourself, but anyway, thank you for attending. And Koei, I guess I'll let you handle any questions that are coming through.
Yes, there's a question. Let me check. I can't find my resource. What should I do?
Right. So if you can't find your resource, as I mentioned earlier on in the talk, we do provide information on where one needs to go in terms of registering those resources. So, for example, if you had an antibody, we do provide information, that the community resources, the antibody registry, and that you would need to register your resource with that community registry. Again, we don't control all the community registries. They're controlled by those community authorities. So, for example, in the model organism section, there are actually more than 20 different community authorities for model organisms, and depending on what type of organism, a model organism you need to register, you'd have to go to the right model organism database. And again, we provide that information for you as to who you should be contacting. And if you are having, if you still don't have sort of information that you think, or if you need assistance sort of in registering your model organism, that's something, or antibody, you can always get in contact with us, and we can help you out as well.
Jeff, hi, this is John Cadiz from the City of Hope. This is very nice work. Thank you. I mean, it's really amazing to see what you guys have done here. I thought one thing that was interesting was your authentication reporting, and particularly when you pull out the problems that you could see with one resource or another. Where do you get these problems, and what kind of problems can you flag? So, for example, I noticed when you were showing antibodies, one flag was a discontinued flag. Is that the only kind of flag that you get? Do you get flags like that's probably something easy that you can do. You can verify with a vendor perhaps, but if there are like user reports that, oh, this doesn't work or something like that, is that another type of flag that you can?
That is another type of flag that, okay, so right now the information that we're pulling again is the information resources we have access to that we can aggregate information from. And so again, for antibodies, that is information from the community authority. For example, if the antibody has been discontinued, for cell lines, from the resources we tie into there, we have some additional information that's provided in terms of, is it discontinued? Some of the reasons, is it contaminated? Is there other issues with the cell line that we're pulling from those information resources? But the question that you had, sort of the second part to your question, user reports, that's something that we're actively looking at, is how we can actually extract that information, for the various resources. So that actually is a sort of an area that we're interested in terms of providing some of that information as well. But that sort of, will be part of hopefully a future release, where we can start aggregating that information.
And Jeff, can I ask you one more question as it relates to your FAIR tool for creating these data plans? Is the idea that this tool you envision helps investigators with their grant submissions in terms of creating plans, or is it, or does it all, or are you also envisioning that it helps people assess the repositories that they've created? I note that I did notice that there was a thing that said request a data repository recommendation. So clearly you can go through and help researchers identify any potential holes. But but in particular, this tool, is this for writing, like a writing aid for investigators?
Yeah, it's a bit of both, right? So in terms of providing the writing aid, right, we need to have information on the repositories themselves. So part of this is the initial step of reviewing the repositories, how fair the repositories are, and other information about the repositories that then can be included in this tool, that when you say, my research study is going to involve this data, that we can provide then sort of the best options, for that data. And then out of that, we do, want to be able to provide then the ability to say that, well, for data management, here are the repositories, that I will be, depositing data into, and provide some information about those repositories and their suitability for the data, so that there is actually, part of the data management plan that is provided.
Thank you, Jeff.
Jeff, I didn't see other questions.
Okay. So if no one else has any other questions, again, if you have any, you're on dkNET and would like to see, any new features or have any comments, feedback, please send those to us. We do have on the bottom of the page, there's a little contact us button that you can always open and send us a note. And other than that, thank you again for attending today's webinar. We'll be making, again, this available on YouTube, and our prior webinars are also available right now. For example, the webinar from last time, which was a webinar on this Hypothesis Center of the Signaling Pathways Project. So again, thank you for attending, and have a great weekend.